Good morning, everyone. Welcome to the day 2 of our virtual conference, A Paradigm Shift in Infectious Diseases Lessons from a Pandemic. My name is Vibhu Jain. I am Head of Commercial Marketing for Asia Pacific for Merck Research Solution Business. I am the moderator of today's module on diagnosis and innovation during pandemic. I would like to introduce our first speaker, Dr. Anne Wyllie from Yale School of Public Health. Anne will share with us about her latest work on SalivaDirect COVID-19 diagnostic test. This test got emergency approval from FDA a couple of weeks ago. Please put your questions to Anne in the chat box. Over to you, Anne.
Hello, I'm pleased to be joining you today and able to share our work on SalivaDirect. SalivaDirect received its Emergency Use Authorization from the FDA on August 15th and has been described as the most unique EUA that the FDA has issued to date. What makes SalivaDirect different is that we haven't developed like a test kit. We're not manufacturing anything, we're not distributing anything other than the method itself. So the EUA that we have allows us to designate other labs who can also get SalivaDirect up and running in their own labs for testing. So SalivaDirect really came about from this main goal that we had to improve testing options. We've been collecting samples from COVID-19 inpatients since the start of the pandemic, where we're enrolling these patients to collect a variety of sample types from them as a way of studying both SARS-CoV-2 but also trying to understand more about COVID-19 disease progression. And right from the start of this, we experienced a number of different challenges. Some of the more major ones coming from the nasopharyngeal swab itself. We experienced firsthand the supply chain issues in trying to get these swabs and having to order in our own tubes and making our own viral transport media and putting these kits together. There's of course the discomfort that it can cause patients, which also carries risk to healthcare workers if the patient is to sneeze or cough on the healthcare worker. We're seeing a lot of testing aversion as people learn more and more about how uncomfortable these swabs are. And I think we also missed out on a number of research opportunities because of either not having enough swabs for the research swab and not wanting to detract from the diagnostic supply chain, but also from the healthcare workers just understandably being concerned about going in for a second swab. So first having to take that diagnostic swab and then having to put themselves at further risk by taking that second swab for research purposes. For RNA extraction, so all the samples that were coming through, again, we're collecting a variety of samples, so being the nasopharyngeal swab, oropharyngeal swab, blood samples, saliva, fecal swabs. These were all being processed for RNA extraction, so we were trying to detect SARS-CoV-2. Regardless of what we were saving them for, we decided that we'd see the presence of SARS-CoV-2 in each of these samples. And for that, we were using an RNA extraction method. Again, we were very keenly aware of supply chain issues when setting this up. So we actually started off by using a kit that wasn't already recommended for the clinical diagnostic testing. It soon became one of the recommended kits. But, so we were aware again of these the supply chain issues. Also, this was sort of the most time-consuming step and also the most expensive part of the SARS-CoV-2 detection. We were testing all of the RNA in RT-qPCR. So we were using the CDC's primer probes for N1, N2, and RP as the human control. We were just ordering these separately from IDT. But again, every sample that we extracted had to be run in these 3 different RT-qPCR reactions, which was also quite a labor-intensive step. So all those issues that we were seeing with the nasopharyngeal swab, that's why saliva really caught our attention. So we were, as I said, we were just collecting these samples, these matched samples from these COVID-19 inpatients. So we had a whole range of matching nasopharyngeal swabs and saliva samples taken from the same patients at the same time points. And as we were watching the results of SARS-CoV-2 detection coming on by, we started to see that saliva was looking quite promising, that we were seeing that it was acting as good as, or even in some cases better than, the detection for SARS-CoV-2 as compared to the nasopharyngeal swab. So for that reason, we decided to keep working with saliva as a sample type and see if we could help validate it and optimize it further for as a testing specimen. So as we were starting that work to validate saliva as a sample type, there were some early preprints that caught our attention where people were taking the viral transport media from swabs and testing that almost directly in PCR. And we couldn't help but wonder, could we do the same with saliva? So it really did stem from quite just a crude test very early on where I was setting up a qPCR anyway and thought, what happens if we just put some saliva into the master mix and get it running? Like, will we see anything? And it wasn't the greatest results, but they were encouraging enough for us to pursue this further. And I think our previous experience with working with saliva as a sample type put us quite in a unique position to be able to develop this. We knew that if we could add proteinase K to saliva samples, this would really just help. It really helps cut through a lot of the mucus and makes it a lot easier to work with. But of course can also break down the proteins in the virus, helping to expose it. So we found that by just simply taking a saliva sample, adding proteinase K, combining it, and in the heat and activation step at 95 degrees for 5 minutes, we could get rid of that whole RNA extraction process. Going on to the RT-qPCR detection, we also realized that if we could just do one simple reaction rather than the 3 in singleplex, that would also save us a lot of time and effort. So we dualplexed the N1 and the RP. So again, we've kept the RP in there as a human control. It's to so that we can be sure that if your sample is negative for N1, you're not getting a false negative due to some sort of inhibition in the sample itself and that the reaction didn't actually work. So targeting N1 for SARS-CoV-2 detection, targeting RP to make sure that the sample was of good enough quality. And the added benefit of taking out that RNA extraction step, it is the most expensive part of the process. It also saves us some time. So in our lab, it can save about an hour. We're thinking that in instances where automation might be in place, this can also save a lot of hands-on time from personnel who have to maybe move plates between different automation instead of just sort of get prepared for RT-qPCR immediately. So one of the other key things that we're doing with SalivaDirect is that we're just having a very, very simple saliva collection process. So we are just simply asking patients, study participants, to simply just pull saliva naturally in your mouth. You're not supposed to cough anything up, sniff anything back. We don't want any of that sort of really thicker, more mucousy saliva. We just want that normal, true saliva that just pulls in the mouth. And what we found is that you can actually just very gently expel the saliva into a tube, and that we don't actually need any preservatives. So there are some quite expensive saliva collection devices that are out there in the market. I mean, these can range anywhere from sort of $7.50, $10, $15, all the way up to $50 per saliva collection device. And, if you wanted to do a lot of repeat or frequent testing, that can greatly add up over just, even a small number of tests. So it's quite important for us to see, how stable SARS-CoV-2 detection was in saliva without any of those expensive preservatives. For our FDA EUA, we performed a Spiken experiment, so we took a very strongly positive saliva sample and we diluted it down in negative saliva so that we would reach 12, 25, 50 virus RNA copies per microliter. We stored each of those aliquots for 7 days at 4 degrees, room temperature, or 30 degrees before extracting them through SalivaDirect and comparing it to the CT values that we had when the samples were processed fresh. And as you can see, we had just very stable detection even after 7 days in each of those conditions. We've also seen this in clinical samples, however. So panel A, the graph in panel A, basically the lines of dots that you see, so each of the dots that are joined by a dotted line represent the same sample. And in panel A, we first of all have the CT values, so the SARS-CoV-2 detection when the samples were processed fresh on the day that we received them. And then we also have the CT values from after a freeze-thaw cycle, but then also after they'd been incubated at room temperature for 5 days or 30 degrees for 3 days. And what we can see that it's very quite stable detection of SARS-CoV-2 just in the saliva, nothing else. And the panel B graph, that just really shows that these were samples that we left out for longer, just at room temperature. And one of the ones towards the bottom there, that's going out all the way to 25 days of just very stable detection just in saliva at room temperature. So for us, this is what's helped us to keep prices down for SalivaDirect and making it just very easy. Many just sort of very simple laboratory plastics that you can use to collect the sample. What was also important for us to establish was that this method would maintain sensitivity. So again, we started developing this really quite early on when, the most sensitive method really that we knew of was RT-qPCR detection. And I think very early on I was absolutely terrified about what a false negative could mean. I didn't really understand or know what was that risk of those very, very low viral loads. So, we worked really, really hard to make sure that when we were developing SalivaDirect, we could get it as sensitive as possible as compared to that method with the RNA extraction. So this is just a graph of some of the clinical samples during our development. And you can see that compared to having RNA extracted, all going through the SalivaDirect method, we did maintain a good level of sensitivity. There is we lost detection of 2 samples. One is near that lower limit of detection. The other one is slightly higher, but that was actually a really good learning point for us as well, because that sample is around 30 CT and then was negative in SalivaDirect. It turned out that it was actually a sample that was really, really quite mucousy. It came from quite a severely ill individual. And just being that quite, it wasn't that normal true saliva. It was very much more, it was more sort of almost like sputum to some extent. And the fact that there was so much more sort of, we found that it was inhibiting the qPCR detection. And I think it was a good learning point for us, and that we needed this just to be good, true, proper saliva. Nothing thicker, nothing more difficult to work with either. This was also our development for the Multiflex Pcr. So again, we'd been using the singleplex for N1, N2, and RP. And actually originally we decided to try and multiplex all 3 of these together into a single reaction. And in the end we decided to take N2 out, just because for us N1 detection for SARS-CoV-2 has been just remarkably stable, very specific, very constant. Whereas we see a little bit of variability in N2, and actually including N2, it led us to a lot more sort of invalid samples. So we'll sometimes often see like a larger discrepancy between N1 and N2 detection. So we just thought for SalivaDirect to keep it more simple, to help improve our results, just to go for the dualplex in the end of just N1 and RP. What's really also quite unique about SalivaDirect is, again, as I said, we're not packaging anything up, we're not selling anything, we're not distributing anything. In fact, we're getting all of the labs to go direct to the vendors. What we've done is at each step of the way, we've validated reagents from different vendors, and we're going to keep expanding on this further. We wanted to do this to hopefully circumvent any supply chain issues, and also to hopefully keep competition between the different vendors, so that you wouldn't see any sort of price increases due to sort of the recommendations that we're putting forward. So through these different combinations, the list price of a SalivaDirect test is about $1.30 to $4.30 for just the reagents. However, a lot of labs already have a lot of existing discounts, or bulk discounts that you can get through the suppliers. So in a number of cases, we think that you can actually achieve about $1 per sample for the reagents itself. Now, we are aware that there are other costs that do need to go on to this. So of course, costs for the logistics, for obviously the personnel to run the test, the facilities to house these tests in, a lot of the regulatory steps as well can be associated with some quite high prices. So we do know that it is going to be more expensive to run SalivaDirect than just this $1 per sample. But I think what we really wanted to highlight is just how cheap it is at the base of it, and therefore, this will hopefully really make the difference. That this should not be a $100 or $150 test, but really more affordable. So using those different combinations, we've validated the limit of detection for each of those various combinations. So this is just showing an example of our limit of detections graphs. So in the instance of these 3 graphs, we were just changing out the master mix. So we were keeping the proteinase K and the instrument constant in this one, and just changing out between the NEB Luna, the Biorad Reliance, and the TaqPath master mixes. Regardless of any of the combinations we achieved, we were getting down to about 6 to 12 copies per microliter, and that's on par with a number of the great PCR tests out there at the moment. And as you can see from the graphs, we were only going down to 6 copies per microliter. We weren't actually going down any lower in the sort of dilutions that we had done. And then recently, especially under the FDA's advice, is that we've made an extra step down to 3 copies per microliter. And actually, when we've been rerunning some of these for our bridging assays so far, we are finding that we can get down for some of the combinations down to 3 copies per microliter. So a good level of sensitivity there. So one of the things that we had to do, of course, was the clinical validation. So the first thing we did was we had the matched nasopharyngeal and saliva samples from COVID-19 patients. So we first had to validate saliva itself as a sample type. So we tested both the nasopharyngeal samples and the saliva on the ThermoFisher TaqPath COVID-19 kit. And again, we had 94% sensitivity as compared to the swabs. So there was one sample that was positive in the nasopharyngeal swab, but negative in saliva. But actually, there were a few samples that were negative by the nasopharyngeal swab that were positive in the saliva. For the clinical validation of SalivaDirect itself, we took those same saliva samples, but then we ran them through the RNA extraction-free method of SalivaDirect, and we compared those results to the nasopharyngeal swabs, and again, we had 94% sensitivity. We also did a quick check on cross-reactivity. So, we were already using N1, so we weren't too concerned about a risk of false positive detection, simply because N1 is already so widely used and tested throughout the country at the moment. And people are continuing to do in silico analyses. It's been tested against a number of different viruses and bacterial species. But we had saliva available to us that was collected prior to COVID-19, so we just thought, well, we have these saliva samples that were collected locally, let's just run them through SalivaDirect and just really make sure that, just as a bit more reassurance that we weren't going to have any cross-reactivity. And again, really nice clean results there. There was no sort of cross-reactivity in N1, so no evidence of false positive detection there. So, in summary, we've received this emergency use authorization. Our main goal originally was to reduce testing costs. We wanted to make testing accessible across the country. We knew that repeat testing was going to have to be required in order to sort of reopen, to get a handle on the virus, and we really wanted to develop a test that could drive these test prices down so we could increase the number of tests in the community. And we also wanted to reduce the implementation time by using saliva and just having this very, very easy to collect sample type that people would be more happily and ready to do so, and just sort of removing that reliance on the supply chain of swabs. So the key points really about our EUA to also note is that, again, this is a protocol. It's not just an LDT, it's a protocol that we are able to distribute to other to high complexity CLIA labs for them to implement this locally. We are going to continue to perform bridging studies, so we're going to make sure that we bridge on more reagents just so that we can avoid supply chain issues. We're going to bridge on more of the PCR instrumentation just so, we don't want people out there having to run out and buy new PCR instrumentation. We'd rather do a bridging study with those labs to get those existing PCR instruments up and running. We also make no profit. Again, we aren't selling this as a method. We're not selling a kit. We're having individuals go directly to the lab, to the suppliers, and order the reagents from there. So for anyone looking for more information on SalivaDirect, we do have a web page up and going. We do also have, which can link to our FDA EUA, the preprint that we've prepared, which we go into some of our results and our findings in greater detail. And we also have a form available for CLIA labs who are interested in applying for authorization to use SalivaDirect. So while we've come up with this method, this is definitely not where we're ending with SalivaDirect. There is such a urgent need. It's been really inspiring. So many people have reached out to us in response to this, and we've had so many labs reaching out to us wondering how they can get up and running. And I think what's also been inspiring about that is because we've made the fact that we've made test prices such an important conversation in all of this, and still labs, knowing how much we're saying that this test should be running through for, labs are still coming to us and ready to sort of work with us and work on bringing their pricing down and help make testing more accessible. It's also been quite equally moving though, seeing the number of schools, local groups, universities also reaching out to us, desperate for testing options and where they can get testing. So we're hopefully going to be on the website updating the list of the CLIA labs that we do authorise to use SalivaDirect. So we're also hoping that that will be a way that these local groups can also connect with labs and find more testing options. We do realise though that we do need better testing options and more testing options, and ideally if we can move away from the lab and from PCR. What's been, I think, one of the most exciting things for me about receiving our EUA is that it's been this unique EUA, and the idea that, there are so many other tests out there in development at the moment, and we are having to see this shift in this way of thinking about testing, where we're going to move away from this idea of diagnostic testing and more into these tests that become important for screening and surveillance. And I'm just hoping the fact that, we've managed to get this sort of more new, more unique EUA means that I'm really hoping that many of the other great tests that are in development at the moment out there will also start being able to get EUAs or get authorised more easily or quickly to expand our testing options further. So thank you very much.
Thanks, Anne, for a great presentation and staying awake for so late in the night for this conference.
Hi, everyone. This is Heather. Can you hear me?
Yes, hey, Heather.
Hi, Anne. Sorry, we're having a little bit of a technical difficulties, but I'm going to go ahead and start the questions here. So the first one that I see coming up is that I wanted to comment on, I like the direction you're taking with the bridging studies. It relates back to a question that came up yesterday in terms of collaborating across the public and private sector. Can you comment, have you been approaching and collaborating and how this may have been impacted by the pandemic?
If anything, the pandemic has just been such an incredible source for collaboration. I've just been so incredibly inspired by the number of research institutes, but also industry partners that just come together and have been so open and transparent with the work that is being done and, the difficulties that we may have been having or suggestions to help each other. I just, this is not really the time that we should be sort of like squirreling away secret research. If we want to sort of get this pandemic under control, coming together to work together and being open with everything we do, that's how we're going to really make progress on that. So that's been really encouraging. And yeah, with the bridging studies, you know, we obviously don't have access to all the different PCR instrumentation people have. So we've been asking people to let us know what they have, and so we can also keep an eye on what other labs have. And I think even today we just shipped some of our samples out for validation. So we're going to have some people validating a few different of the other, like the Quant Studio machines for us, just so that we can get them onto the EUA to help other labs get up and running as well.
Oh, that's fantastic. Just inspiring. I love the approach. I know that we're getting questions that are coming in. Really, I love this just opportunistic, you know, everybody wanting to get forward faster. In terms, I have another question here too. In terms of, can you comment on your plans to increase the throughput? So if we're looking at, you know, high throughput, 384 well, can you maybe comment? I know you have different platforms, but comment on, you know, your future plans there.
Yes, indeed. So some of the samples that we've sent out for the validation and the bridging studies today, they are for 384 well platforms, because again, that was something that we didn't have, but we know that others do. We're also reaching out to labs. We know that there's a number of automation systems out there. You know, we're a tiny little research lab. You know, we've been doing everything manually by, you know, manual pipetting, but it'd be really great if we could transform this onto, you know, some of the Hamilton robots, just to really increase sample throughput. So again, looking to work with people on that.
Okay, so we're getting, that's fantastic. We're getting a couple of questions in here in terms of, I'll take it in two parts. So I'll ask the first one. So is there any pre-treatment in terms of the saliva as part of the process, or is that direct?
I mean, in a way, the only so I guess yes and no. I mean, the only pre-treatment per se that we're doing is that simple treatment with proteinase K. Proteinase K is just something that we've found just really, it really just helps cut through any of the slightly thicker samples. You know, it helps us make the sample easier to prepare. It also probably helps us start breaking down the virus and opening that up. And then that's just followed with that simple 5-minute step at 95 degrees to essentially inactivate the proteinase K just before that goes into RT-qPCR. I know some labs have been a bit worried about opening the samples and so looking for heat inactivation steps. You know, we're doing this in a BSL-2 lab, so that's not been too much of a concern for us. But we are aware that others are worried about this, so we are going to sort of also validate on some of those pre-treatment heat steps that some people have been using just to show that this doesn't have an effect on downstream detection.
Perfect, thank you. Okay, then I'll ask the second part of that question, and it's really a pivot in terms of the previous treatment, but more of an I'm going to struggle with that word, but what else, you know, is there a limitation in terms of the methodology? They're asking about, you know, could you use it for drug testing, like drugs of abuse, you know, other infectious diseases? Are there limitations that you know, can think of, or is maybe comment about, you know, what you kind of see this path forward in the future?
Yeah, so indeed, so SalivaDirect was really developed for SARS-CoV-2 detection. So I'm not actually in the, I haven't done too much else in terms of like drug testing or anything like that. So I'm not sure what would need to be done for that. But what we do hope is definitely to expand this. We would like to expand this for detection of other respiratory tract viruses. So especially, you know, going into flu season, the fact that this is a dualplex PCR, if we could get some primers and probes in there to make it multiplex for sort of influenza A and B or maybe other common viruses, so that we could, you know, test straight away for, is it SARS-CoV-2, is it influenza, is it something else? Yeah, we're looking to expand it for that.
Okay, thank you. I think we're just about time with at our time for Q&A, but what I'd like to do now is just thank you so much for joining us this afternoon. We've really enjoyed your presentation and as part of the third theme. So thank you, Anne, and then we're going to go back into our other presentations.
Thanks for having me.